If the first line of defense or our surface barriers are breached, well then there is a second line of defense, which are called internal defenses. So now the microorganisms have reached our tissues, either through the skin or through the mucous membranes. However, our second line of defense are there to protect us, and they're listed here, which we're going to go over individually, all of these items. But before we do, we should mention that we have the ability, or our second line of defense, to identify potentially harmful substances. Because some cells in our second line of defense, specifically macrophages, they have pattern recognition receptors. That means they have the ability to recognize certain structures or antigens on microbes like bacteria or viruses or fungus, so they know that they're not human cells, but they belong to some invading microorganism. So one class of pattern recognition receptors that is studied frequently are something called TLRs, toll-like receptors. And we have 11 different types. For example, macrophage can identify the bacteria causing tuberculosis and act to destroy it, or salmonella has a specific protein on its surface that toll-like receptors on macrophages can identify and attack. So let's begin our second line of defense with our phagocytes. And these are cells that we've seen before in previous chapters that will ingest or eat foreign invaders. So our phagocytes in our blood are neutrophils. Those are the white blood cells or leukocytes that we have the most of. They upon contact with infectious material, they will become phagocytic, and they will actually destroy themselves in the process. And also, there's macrophages that develop from monocytes. Monocytes are white blood cells in the blood, but once they enter the tissue, they become macrophages, which are free when they wander through tissue spaces, especially in areolar connective tissue, or fixed macrophages, permanent residents of some organs. And there's some examples here. Definitely one we've seen last semester, microglia, which are phagocytes in the brain, which is a type of neuroglia in our nervous tissue. And if we look at the events of phagocytosis, we can see a pathogen being ingested by a macrophage here, and once it's ingested, it will join up with a lysosome, and they come together, and now we have something called a phagolysosome, which the lysosome contains toxic compounds, and when they join together, those toxic compounds will destroy the pathogen, and sometimes the pathogen is released through exocytosis. So our phagocytes, like neutrophils and macrophages, are able to ingest the pathogen because they look at the carbohydrate signature. So these are on the surface of the plasma membrane, of the cell membrane of the pathogen. So those carbohydrate signatures, that sugar coat, is what the phagocyte recognizes as foreign and invades them. But many bacteria have some capsules over their surface, so they're hiding their signature. So in that case, we have something called opsonization. Our immune system can coat the pathogens that have capsules with opsonins. And these are complement proteins, which we'll discuss a little later on, or they could be antibodies. And all of a sudden, these opsonins act as handles. So phagocytes can now grab onto them and ingest them. And when pathogens are ingested, like we saw in that image earlier, it joins up with a lysosome that have toxins to destroy the pathogen. But sometimes the pathogens aren't killed by the lysosome that joins up with it. So in that case, we need some other cells to help out in our immune system. They're called helper T cells, and we'll look at T cells later in the chapter. But T cells trigger the macrophage or activate enzymes inside the macrophage to produce something called a respiratory burst when the lysosome cannot join up and destroy the pathogen. So the respiratory burst will, and here's what that includes: releasing cell-killing free radicals, producing oxidizing chemicals like hydrogen peroxide, increasing pH. So all of these things are occurring in this respiratory burst to destroy the pathogen when the lysosome toxins itself cannot. That's in a macrophage, but if we're in a neutrophil, we have something called defensins that we see defensins that we've seen in a previous chapter that help pierce and destroy the pathogen.